Programmed cell death, also known as apoptosis, is a highly regulated process that leads to the orderly dismantling of a cell. It's crucial for development, tissue homeostasis, and removal of damaged or infected cells. While the initiation of apoptosis can occur through different pathways, the execution phase shares common steps. Here's a breakdown of the general steps involved. It's crucial for development, tissue homeostasis, and removal of damaged or infected cells. While the initiation of apoptosis can occur through different pathways, the execution phase shares common steps. Here's a breakdown. This is the stage where the cell receives signals, either internal or external, that trigger the apoptotic program. There are two main initiation pathways. External signals, such as the binding of death ligands, for example FA's ligand or TNF-alpha to death receptors on the cell surface, activate a signaling cascade. This leads to the formation of the death-inducing signaling complex, or DISC, and the activation of initiator caspases, specifically caspase 8 and 10. Internal cellular stresses like DNA damage, lack of growth factors, or endoplasmic reticulum stress trigger this pathway. These stresses lead to changes in the mitochondrial membrane potential and the release of pro-apoptotic proteins such as cytochrome C into the cytoplasm. Cytochrome C then binds to apoptotic proteas, activating factor 1 or APF1 and procaspase 9, forming the apoptosome, which the initiator caspases such as caspase 8, 10 or caspase 9 activate executioner caspases like caspase 3, 6 and 7 through proteolytic cleavage. Executioner caspases then dismantle the cell by cleaving various cellular substrates, including nuclear lamins, which lead to nuclear shrinkage known as pycnosis and fragmentation or karyorrhexis. They also target cytoskeletal proteins, causing cell shrinkage and the formation of membrane blebs. Additionally, the SAD, or caspase-activated DNase inhibitor, is cleaved and inactivated, allowing CAD to degrade. As the execution phase progresses, the cell undergoes characteristic morphological changes, such as cell shrinkage. Cell shrinkage means the cell volume decreases. Membrane blebbing is when the plasma membrane forms bubble-like protrusions. Chromatin condensation or pycnosis is when the nuclear chromatin becomes dense and compact. Supernuclear fragmentation, karyorrhexis. Nuclear fragmentation, known as karyorrhexis, is when the nucleus breaks down into smaller fragments. Superformation of apoptotic bodies. Formation of apoptotic bodies occurs when the cell fragments into membrane-bound vesicles containing cytoplasm, organelles, and nuclear